Hello, and welcome to Sidhu Academy. It's great to have everyone here and ready to learn. Well, my name's Hritik. You can call me Hritik Sidhu, Mr. Sidhu, Hritik. Just say my last name Sidhu, anything you really want. Today's lesson is one that I find very important in terms of domestication. Domestication is a state where an animal is taken from a wild state and adapts to close association with humans and often to the benefit of humans. When we think about domestication, we view it as a process that takes an immense amount of time, and it has happened to many species many years ago. We often hear stories of wolves being tamed through sharing of scraps foods with prehistoric humans. These wolves were further domesticated through selective breeding, and over hundreds of years and thousands of generations to become different species of dogs. While we know dogs are man's best friend, a similar creature, the red fox, also known as Vulpes vulpes, which has been tamed by different humans around the globe, is one species that is being looked at in a study for domestication. Let me get one thing straight. Tame does not mean domesticated. Taming is done through behavior modification of a single individual of a species, while domestication is the permanent genetic modification of a lineage of a species, often leading to a heritable trait benefiting humans. Many think that the domestication of the wild fox would take hundreds of years. However, an experiment done in Russia has shown otherwise. At the Institution of Cytology and Genetics of Russia's Academy of Science, Dmitry Belyov, a fan of Darwin's work, was fascinated by domestication syndrome. If you were to ask Darwin, what is domestication syndrome? He would say, "What the hell are you talking about?" Because the term was not coined by Darwin. However, Darwin describes multiple morphological and behavioral characteristics that were present in the majority of domesticated populations. There's a variety of characteristics, but the main ones include tameability, change in ear form, tail form, skeletal form, often in the facial form, and coat color. There's also shown to have a loss of reproductive seasons, such as we see in chickens. Dmitry hypothesized that our ancestors first selected for tameness of an animal to start domestication. He hypothesized that tameness is related to the domestication features that we see today. Dmitry used silver foxes, a coat variant of red foxes, with a 1.2 kb deletion mutation at the ASIP gene, diverging from the wolf lineage 10 to 12 million years ago. The experiment started off with 30 male foxes and 100 female foxes, where foxes were selected based on their behavior when a human is present. To ensure these foxes were only selected due to tame behavior caused by genetic selection, foxes were kept in cages separate from each other and had minimal contact to humans. The top 10% of tame foxes were allowed to breed and create future generations. Within 6 generations, around 6 years in breeding seasons, foxes were able to lick the hands of experimenters, were able to be picked up and petted, whined when humans left, and wag their tails when humans approached, acting like dogs extremely fast. In less than 10 generations, foxes started to have increased phenotypes of domestication syndrome characteristics, such as floppy ears, curly tails, and they started to produce mutt-like fur patterns. Over the course of the experiment, the facial features had also changed with more juvenile facial features, such as rounder and shorter and more dog-like snouts, also including chubbier limbs. In the late 1960s, to complement their findings of physical change due to tameness, the experimenters had created a lineage of aggressive foxes, where foxes were selected for aggressive behaviors rather than tame behaviors. To compare both aggressive and tame lineages, the researchers had allowed an unselected population to breed without human interference. As we can see there are physical changes, we must also see genetic changes, as domestication is a permanent genetic change. We must look at the genomes of red foxes to determine the changes that have occurred due to breeding of different strains. Anna V. Kolokov and her team did a genetic analysis of each population in the experiment and compared them against a fox genome. the genome of 10 foxes from each of the three populations, those being tame, aggressive, and conventional meaning the unselected population, were sequenced. These genomes are ran through Structure. Structure is a program that can identify subsets of a whole sample by detecting the allele frequency differences. Structure can assign individuals of a subpopulation based on the analysis of those likelihoods. So, we can use this program to measure the divergence in each population. Below, I would like you to look at the structure graph. The graph is labeled structure, but it's also labeled 1A. This graph was taken from Anna Kolikov's research. Each bar of this graph is an individual, and each color is the existence of a cluster of genes in that individual's genome. The length of the colors is the estimated proportion of the individual's genome. The assumed number of clusters, K, is indicated on the y-axis. In this example, we can see how aggressive species are less divergent from conventional foxes, but tame species are highly divergent from conventional and aggressive counterparts. This shows us that there is a genetic change, and quite a big one. The structure analysis allows us to see genomic regions that are subjugated to selection in both aggressive and tame populations. We must look at heterozygosity to determine if any of these alleles have become fixed in these populations. Currently, I would like you to look down below at the graph labeled 1B, which has the FST values and heterozygosity values of each population. Heterozygosity was calculated through the use of 9,151 windows, which were around 500 kilobytes each, and this spans the whole genome. In this image, we can see FST and heterozygosity of each population. We have the top FST being for comparing the tame and aggressive. We have the heterozygosity for tame in the top. We have heterozygosity for the middle, which is aggressive. And we have heterozygosity for the bottom, which is for conventional. The population-specific cutoffs were a p-value of 0.001, highly specific. In each of our examples of heterozygosity, we can see fixing of alleles. In the tame species, we have 96 low HP windows. In the aggressive population, we have 60 windows that are low heterozygosity. In 14 windows on the conventional, we have low heterozygosity. We can see that in each of these populations, there's fixing, and there's more fixing in both the aggressive and in the tame population compared to the conventional population. In total, 138 annotated genes found in the HPT windows, 159 in the HPA windows, and 151 in the HPC windows. Using FST, the researchers were able to identify regions of extreme difference between the fox populations. Only 3% of the windows between the tame and aggressive populations had an FST value of 0.458 or higher. 106 windows in the analysis of the tame and conventional, and 1 window in the analysis of aggressive and conventional. Using this information, we can see that there's an extreme difference in the windows of tame and conventional. Using the information of extreme differentiation windows, the researchers were able to find 103 regions in the fox genome that were positively selected in terms of tameness. A previous study found and identified 9 fox loci that influence behavior. These 9 loci were first discovered through the use of quantitative trait locus analysis. The QTL uses linkages between physical and genetic changes to search for a gene that influences a qualitative trait, such as height or skin color. Anna V. Kolkov was able to compare her 103 regions to the QTL data. She found that 30 of these regions overlapped with 5 of the fox behavior quantitative trait loci. These 30 regions are the most likely targets for selection in terms of behavior. Using region 94, we're able to see a significant FST value between tame and aggressive. This region is also part of the SORCS1 gene, which has been identified for the behavioral phenotype DPC1. Higher DPC1 means foxes will solicit observers' attention after an interaction. Lower DPC1 has shown that foxes will avoid observers after an interaction. So higher DPC1s are basically attention seekers. The function of the SORCS1 as a global regulator of a synaptic receptor supports the role of SORCS1 in the regulation of behaviors between tame and aggressive foxes. Outside of the context of behavior, researchers were able to find genes that were fixated in both tame and aggressive lineages. Using the HPT, which is the heterozygosity of tame lineages, we are able to see a window of enrichment for single guanine insertion binding and damaged DNA binding. Therefore, regulation of DNA stability might increase tameability in some way. Using HPA, which is the heterozygosity of aggressive lineage, we are able to see the enrichment of genes associated with immune response, meaning that immune response and immune genes play an important role in the selection for aggressive foxes. Kolkov and her team are able to show us a fox genome of both aggressive and tame lineages. We're able to see different frequencies of alleles and fixation of alleles, which can show us alleles that are selected for. However, we're not able to see the changes in physiology, morphology, and reproduction due to different mechanisms, such as phelotropy, random fixation, trade-off between biological systems, and gene expression, especially through DNA methylation. To learn more about domestication in terms of domestication syndrome, we must look at changes in hormone levels. A trend that is observed in our tame fox population is the reduction of stress hormones. The levels of stress hormones, including cortisol levels, were decreased by almost half in generation 15. The adrenal gland had also become smaller with further generations, leading to decreased testosterone, adrenaline release, and androstenedione, which can lead to more juvenile complexions. To look into the increases and decreases in hormones, Zhu Wang studied the gene expression in 2 regions of the brain. Using 12 aggressive and 12 tame foxes, he studied the prefrontal cortex and the basal forebrain. Zhu Wang and his team found that the most significant change was in 2 genes, the DKK-L1 gene and the PCDHGA1 gene. DKK-L1, also known as Dickoff-like protein 1, has a signal transductor activity and interacts with non-clonical WNT pathway. Overexpression of DKK-L1 in the ventral hippocampus, not the prefrontal cortex, has been associated with increased susceptibility to social stress in mice, meaning that foxes with increased DKK-L1 are more socially aware. CDHGA1, the protocatharin gamma subfamily A1 gene, encodes for a neurocatharin-like cell adhesion protein which plays critical roles in establishment and function of specific cell-to-cell connections in the brain. In other species, its expression has led to phenotypical changes in behavior. Serotonin and serotonin metabolite levels have been found to be significantly elevated in tame foxes compared with aggressive foxes, which is similar to most mammals and invertebrates that have been domesticated. In tame foxes, serotonin receptors 5A, 3A, and 7, and as well as a pair of downstream signaling genes, DUSP1, and AKT1 were expressed to increase serotonin signaling, which means that they were both in gene expression and allele frequency higher in tame foxes compared to aggressive foxes. We can also see this trend of gene expression and allele frequency increases in the glutamate receptor signaling pathway of the foxes. The expression of GRIN2D was elevated in tame individuals in the forebrain, and GRM3 showed significant frequency changes in both aggressive and tame populations. Downstream signaling genes of the glutamate receptor pathway, ITPR3 and ADCY7, were significantly upregulated in tame foxes. Downstream signaling genes of the glutamate receptor pathway, ITPR3 and ADCY7, were significantly upregulated in tame foxes. It might just seem like I'm spewing out a bunch of genes, but these genes actually show parallels to many other domesticated animals. In genes of glutamate receptor signaling, we can see that dog genes had also gone through selection of glutamate receptor signaling, such as GRIA1 and GRIN2A. And similarly, in domesticated cats, we can see two glutamate receptor genes, GRIA1 and GRIA2, which are also under positive selection. And a recent sweep done in rabbit shows a selection of GRIK2. We also find that ITGA6 was also found to have significant allele frequency changes in dogs and other mammals as well. Comparing with other species allows us to see these micro changes that lead to domestication of animals from the past. The experiment shows us an example of domestication in our modern world. However, we should not be so quick to trust everything that we see. Many of you thought in this experiment that the foxes were wild. However, the foxes were fur trade foxes from Prince Edward Island in Canada. These foxes had been bred for variations of coat color and patterns since the 19th century, or the 1800s. We cannot be sure if the selective breeding of coat color had an effect on the gene pool of the fox population. The experiment had always said that they only selected for tameness, but the farmers had selected for coat colors beforehand, which could have helped domestication syndrome traits emerge, such as mutt-like coat colors. A small starting population of only 130 individuals can make experiments highly susceptible to allele shifts and strong founder effects. This can cause random selection of traits rather than based on tameness alone. Like I said before, coat colors could have been due to a random selection rather than founder effects. We also need to look into experimenter bias, as many traits described by experimenters were found in wild populations as well. Droopy ears were found in wild populations of PEI. Coat colors that Balayev described were also found in wild populations. And a change in seasonality was reported, but there's no increase in fecundity. While traits of domestication syndrome have been observed, we should not treat this experiment as a wild to domestication experiment. These foxes had over 150 years of selection, then had their selection changed to tameness instead of coat color. Now, 71 years later, domesticated foxes are able to pick up social cues from humans and have social tolerance. While this might not be the perfect study in terms of domestication syndrome, it sheds light on what genes have to be upregulated, downregulated, and selected for when it comes to domestication. A further study of neural crest cells, which are the stem cells that encode tissue for domesticated syndrome traits such as shortened snouts, curly tails, white spotted fur on coats, and floppy ears, would help shine light on whether this process of selecting for tameness leads to domestication syndrome traits. Several of these genes that manifested significant allele frequency changes in our tame foxes may play a role in the neural crest cell fate. These include the WNT signaling, which plays a key role in the initial neural crest cell differentiation. And both WNT3 and WNT4 in the foxes have more than one SNP with significant allele frequency changes.